Hello, good afternoon. Thank you all for joining this webinar. This is the second of 4 webinars focused on Charcot-Marie-Tooth neuropathies. This is organized by the European Reference Network for Rare Neuromuscular and Rare Neurological Diseases in collaboration with the European Academy of Neurology and the European CMT Research Association, ECRA. So today we're very happy and honored to have Andrea Cortese with us. Andrea Cortese is a professor at the Department of Neuromuscular Disorders at the University College London at the Institute of Neurology, with a part-time appointment also at the Carlo Pesta Neurological Institute in Milan. He is famous already for his great discoveries in CMT genetics. His research focuses on the discovery and modeling of novel causes of CMT, with particular interest in repeat expansion disorders and conditions caused by variations in non-coding DNA. His main impact in the field has been the identification of the RFC gene repeat expansions that cause the canvas syndrome, and also the genetic cause of the sorbitol dehydrogenase, the SORD CMT neuropathy. Both of them have been now recognized as common causes of inherited neuropathies, and his work has also led to the development of clinical trial for SORD CMT, which is in progress. So really great impact in the field. So we're very excited to hear Andrea's talk, and thank you, Andrea, for joining. And we look forward to your talk about progress in genetic diagnosis for neuropathies. Thank you,
Thank
Andrea.
you so much, Cleopas, for the invitation, and to the organizer. It's a great pleasure to be here. I know there's some people I know online. I appreciate I don't see them, but saying hi to them, and to whoever was able to connect in the middle of the day. I continue to share. I just want to make sure you now see my slides.
Yes, everything looks good.
Perfect. Okay. So I'm gonna tell you a little bit about what Cleopas has said, what I've been doing, what we are doing at the moment at UCL, and in also starting to implement similar approaches in Milan. And I'm really gonna give some example of the more recent use of long-read sequencing, other novel sequencing technologies in neuromuscular diseases, and with a focus on advancing genetics in CMT. I hope it moves. Yeah. So, but to speak about long read, let's see first where the current technology stands in, at least in the UK. And the current gold standard is a short read whole genome sequencing. Every patient with a suspected genetic condition gets whole genome sequencing in the UK as first test, with some rare exception, like testing duplication for PNP22 in demyelinating CMT cases. But you see that despite, best efforts in neurology, but really across medicine, these are all different specialities, the diagnostic yield is still unsatisfactory. 2 cases out of 3 remain without molecular diagnosis after short-read whole genome sequencing. Now, the percentage is slightly higher. Diagnostic yield is higher when you have a metabolic marker, when you have an early onset in parents to perform a trio available, but still not quite there. So where is this diagnostic gap in inherited genetic conditions and inherited neuropathies as well coming from? Well, we realized that over the years, since the introduction, in particular, whole exome sequencing, there's been really a boom, an explosion in the number of genes that were identified. But papers tended to report novel genes with decreasing number of families and patients. And if this was to continue, probably, in terms of code environment, we may find genes which are disease causes in a family, but it's private. So very hard to prove this is real without replication in independent families. I think that, a few years ago, I would have said, I don't believe much diegetic inheritance in Mendelian disease, but I think I've been proven wrong. I think there's good example, particularly for the muscle field and ataxia field, where tied in with SRPK X-linked, by the way, this one causes myopathy. I think FSHD2 at the end of the day is a diegetic disease. In distal myopathy in Finland, the interaction TR1 and sequestrosome mutation, and a finding from Milan Besta from Stefania Mag a couple of years ago was an interaction between SCA17 intermediate alleles and STUB1. So it might be that this is still an underexplored cause of Mendelian disease. Of course, one of the biggest challenges, and I'll come back to that at the end probably, is the functional annotation and interpretation of coding, but particularly non-coding variant. Coding to the coding is 1 to 99 in terms of size in the genome. So a lot of non-coding variant, we don't know what to do with. But my presentation is going to probably focus on the missing heritability coming from limitation of the current sequencing technology, short read. This is clear to everybody when we look at exomes, because we're looking at, say, just 1% of the genome. We're ignoring 99% of it. But also, short read whole genome sequencing struggles when it comes to repeats, so short tandem repeats, but also repeated region, duplication, deletion, inversion, and in general structural variant, particularly if they involved similar sequences. And so to guide you where we're going, I'm just going to do a brief introduction, what technologies these technologies are, but it will be a repetition for some, many will be familiar. And then I give some example of their use in the field of genes, pseudogenes, or structural variant, repeat expansion, and then for functional validation of mainly splicing variant. So short read, long read, well, the name is self-explanatory. The difference in the length of the read, 150 base pair, short read, kilobase is long read. And while short reads need to be aligned, mapped to the reference genome, and if a read could align alternatively to this location, and this one, because they are similar in the reference, but the read cannot be really aligned because the system doesn't know where to put it, and it will be discarded. So there will be gaps left. Long read, thanks to the read length simply, will provide a much more even coverage across the genome, and will add the portion anchored to unambiguous region would help to also sequence the more ambiguous repeated region. There's two main technologies available, Oxford Nanopore. Sequencing is provided by changes in current induced by the passing of the single DNA molecule through a pore. When there's an A or a G, you got changes in the current, and this is decoded into nucleotides, and there are various sizes, and one is really portable and very easy to use pretty much everywhere. The other main technology is PacBio. This is a bit more similar to Sanger sequencing. Illumina is sequencing by synthesis. So when you add the nucleotide, there's a polymerase. Whenever a nucleotide is added based on the template, which is your DNA, well, it will emit a fluorescent signal. Now, the difference here is the long DNA molecule is ligated as a circle, and it passed through the polymerase many times, increasing accuracy of the nucleotide calling. The last technology we're using is optical genome mapping. This is not sequencing. This is actually some sort of imaging of the DNA, and it's not that different from Sargent blotting arrays. That it provides that kind of sequence at high throughput level. So here what happens is the long DNA molecule are fluorescently labeled using repetitive palindromic sequencing, and those are spaced in the genome, and the distance allow us in a way to know where these regions are. And so you get all the molecules tagged, then you image them, you see the green dots, you measure the distance, and you say if there's a 3 dots, 4 dots here, and then another 2, 3, 5, you say, oh, we're looking here at MPZ, for example. We know that this fluorescent signature. And then if you imagine that these 3 dots and these 2 fluorescent dots are closer than what they should, you can imagine there is a deletion here. If they're farther apart, there might be an insertion or a repeat expansion. And this is how it works at a genome-wide level. And here's a comparison. We processed a sample with the 3 available technologies. The cost of long-read is still quite high, $2,000 approximately. Long-read optical genome mapping is more $800. The long read and short read, they produce a lot of data, so there's much higher cost in terms of storage, even more for long read if you want to retain methylation. Optical genome mapping is smaller. In terms of single nucleotide variants, I would say short read remains for the unambiguously mapped gene regions the reference techniques, but I would say the long read is catching up. This is where I'm pointing. And but clearly the advantage of long read is when it comes to insertion and deletion. Many more are picked up compared to short read. And this is also an excellent method to test the repeat, providing information about their accurate size, while the size from short read is mostly estimated, and also its sequence. While in optical genome mapping, repeat expansion will be seen as an insertion, really. And long read has 2 advantages. It also provides information about phasing. So if you want to know the phase of the variant in a recessive gene, with short read, you usually need parents. While for long read sequencing, the reads are long enough to cover the entire gene and show that in one read you have 1 mutation and on the other read you have the 2nd mutation. So it really resolves this impasse and provides a final molecular confirmation. While, as said, OGM does not provide sequence information, only structure. And it's fair to say that our knowledge of the DNA and our genome has tightly followed the introduction of different technologies, from Sanger sequencing with the Human Genome Project, arrays with all the GWAS association studies, then the exciting time in CMT and in other rare diseases of exome sequencing, many more genes identified and genomes, and more recently through the integration of long-read and optical genome mapping, the entire final sequence of our genome was achieved. And this was just a few years ago, 3-4 years ago. The complete sequence of the human genome was published on an article on Science. And in general, we gathered 5% extra information, so 5% of the genome we didn't know of. But if you look what's in this 5%, it is mainly repeats and duplicated regions. And I'm going to start with the duplicated region because knowing what is normal in the population and is the prerequisite to interpret pathological variation. And I'm going to give a particular example of repetitive region, which are pseudogenes. So pseudogenes are copy of usually non-functional in most cases, or at least tend to be considered non-functional of coding genes, which can be reinserted in a way, duplicated in our DNA by either insertion of the transcript itself, and this is a process pseudogene, there are no introns, or by duplication. So this parent gene and this pseudo gene, which over the years, if not necessary, is not under selective pressure, it will accumulate in yellow non-coding variants, which will inactivate it. Still, it will be a pretty similar copy to this one, and we know that similarity in genetics is what drives or helps recombination. And I think in the literature there are quite a few examples of gene conversion, duplication and deletion involving genes and their pseudogene, but there are rare examples, very few, about inversions between genes and pseudogenes, with Hunter disease and hemophilia being probably the most common. So here you see pseudogene gene in opposite orientation. There's an inversion of the first part of the two genes, with the pseudogenes moving to the gene, inactivating it, and part of the gene moving, switching to the pseudogene, which will still remain inactivated because of the other variant. So the effect here would be loss of function of the parent gene. And the reason we speculated why these are not really picked up, why they're so rare, is really limitation of the technology, because they're so similar, the two, that the short read would not be able to tell you whether this recombination here is there. There's not enough to cover the actual breakpoint, while long read are able to have enough nucleotide to tell you, for example, here that this part here and this part here of the gene. I hope you see my pointer.
Yeah, just fine. Yeah.
I can also get a better pointer. No, this is this is wrong. I wanted to yeah, this is okay, fine. This is I'm just gonna well, anyway, so the read is long enough to cover the breakpoint. I have to change now this weird marker I've selected because I cannot move to the next slide otherwise.
Maybe you click on the slide.
Yeah, I'm trying to click on the next slide, but it's let me see if I can. You see, it's getting me to draw on the slide. I wanted to get a pointer. I apologize, everybody. So stamp, draw, text, and mouse. So I want the mouse, but it's still not working. Let me get out of the presentation if you don't mind.
Sure.
Apologies, and let's see if I start again. Apologies for this inconvenience. Continue to share. Okay, let's stick to the mouse. Okay, let's not risk it again. So, right. So we asked ourselves, could this potential inversion between gene and pseudogene in our genome be more common? So what we did, we took it a genome-wide level, and we looked at all possible pair between genes and pseudogenes in opposite orientation, so permissive towards inversions, and we identified 270 parent gene and 400 candidate gene pseudogene pair, which may, because of their proximity and orientation, undergo inversion. Interestingly, 46 of these parent genes are already homing genes. So finding this inversion may be relevant to the diagnosis of these patients. And in the meantime, the 1000 genome project, long-read data were released. So we were able to benchmark our hypotheses and with a quite complicated bioinformatic method, test for the presence of gene pseudogene inversion. So this is not trivial, but what we realized that 37 of these roughly 1000 individuals did bear gene pseudogene inversion in their genome, which is about 3% of the population of the alleles. And very interestingly, none of them, apart from one, was picked up from short-read data on genome. And again, something that will sound familiar as a name to some of you, the top candidate, the one with more inversion, was SORD. Now, SORD, some of you may know, is probably the most common cause of recessive CMT. We identified it during my postdoc in Miami, in Stefan's Zuckner lab. And there's a common deletion in exon 7, which inactivates the gene. The gene codes for an enzyme. This is really a metabolic disease, sorbitol dehydrogenase. If you have two mutations, you stop the polyol pathway from progressing and you accumulate sorbitol, which is toxic. And we thought, this was a great example of how a discovery, which was kind of delayed, really for the similarity between gene and pseudogene and challenges for long-read sequencing to distinguish the two. But once identified, and with the excellent sorbitol biomarker, a phase 2-3 study started fairly soon. Now is in its open label extension. We're really looking forward to hear about final analysis and data with a small molecule govorestat, a type of aldose reductase inhibitor with limited liver and renal toxicity, able to downregulate aldose reductase and hopefully reduce the influx in the pathway, which is something that has been shown, but hopefully also make patients better. But going back to genetics, and also within the frame of the trial, we identified 3 patients that had a raised sorbitol, and to me, increased sorbitol to these levels, 10 microgram per liter, in controls is almost 0, means they have SORD CMT. But with short-read whole genome sequencing, only one variant could be identified. So we speculated, could there be something there which is not picked up by short-read technology? So we performed long-read sequencing and optical genome mapping, and I'll show you the plot. And we identified in the 3 of them an inversion as the one that I was showing you before. Like if you think here, this is our gene, this is our pseudogene, and I'm kind of flipping them over here so that our SORD starts with exon 1, 2, 3, 4, and then you got 3, 4 exons from the pseudogenes, plenty of nonsense mutation, which will undergo nonsense mediated decay. So no viable protein from this one, and if on the other allele, as in this case, there's a coding variant, well, you'll have no sorbitol dehydrogenase. And it's interesting to see how, looking at SORD CMT patient with the inversion and a control here, you see that short read down here of the region of the inversion is identical to what you see in a control. So there's no difference. While the long read clearly show the break point with this guy here, SORD going to SORD to P, and here the same, and clearly identifying the break point in the 2 gene and pseudogene, suggest you cannot sequence the whole inversion, it's too big even for the read length, but you identify the 2 breakpoint and you can infer there is an inversion between SORD and SORD2P. You can confirm it either faster and with the optical genome mapping, the ultra long molecule of genome mapping, you really get the impression here, how the inversion works. So this marker here goes here, and that's why you got this inversion plot, which confirms the presence of the inversion in all cases. Those technologies are expensive. We tried to develop a PCR assay across the breakpoint, which allowed us to identify many more cases. And indeed now, SORD inversion, invisible to short read, are the 3rd most common variant causing CMT SORD, after the common 757 deletion and this missense mutation in position 153. So not something that can be forgotten in a diagnostic pipeline. And this is another more recent case from the US, tested negative for our inversion, still with one nonsense variant in one allele. By performing long read and optical genome mapping, we identified a new breakpoint. So there might be more than just one recurrent inversion site between SORD and SORD2P. So really, this is one that may need long read on a longer term. And finally, we asked ourselves why SORD2P are so prone to these inversions, and we turn back to chromatin. So it's interesting to see that on the chromatin plot, here you see SORD and SORD2P, and red in this plot means this region and this region here are in contact. And if you look, it's like a stripe, like if the two were linearly distributed one along the other. And I think the proximity of the two, the similarity of the two is what really drives the recombination, which could be simplified as a flipping of the tip of this loop from the gene to the pseudogene. And we think that this is a mechanism that might apply to other gene pseudogene pairs, and we looked here at genome-wide level at in the presence of inversion and compared it with the presence of chromatin contact, and we clearly see an enrichment in genes undergoing inversion with their pseudogene in terms of chromatin contact compared to the ones that are distant matched but don't have this gene pseudogene structure. So we think the proximity in IC, the contact, does favor this inversion at genome-wide level. Another field which has highly benefited from the use of long read is, without any doubt, the field of repeat expansion. So over the years, several repeat expansion have been associated with common mainly neurological conditions, including the most recent one in the field of ataxia with Canvas, FGF14, not present here, but definitely top discovery from Bernard Bray and Stefan, and other genes. And we also have a review which really focuses on the most recently identified one in epilepsy, ataxia, neuropathy, Canvas, Parkinsonism, muscle disease. And I will focus really on these two, Canvas and muscle. But to take a general look, I think that this novel repeat taken as a group, they're teaching us something. So they're mostly located in the non-coding DNA. Sometimes they flank ALU element, were only found in higher primates and human. So it's something that's not very easy to model in animals like mice. They're very large, so difficult for short read, but perfect benchmark for long read. The neurologists need to think about them, know them, because they don't always have a family history. Either they're sporadic or they got a very strikingly variable penetrance. But most importantly, I would say that the pathogenicity depends on both size and sequence. And I think the repeat motif of this repeat expansion is particularly crucial, as I'll show you in the next group. And we'll start with the CGG repeat expansion group. So CGG have been associated with a number of phenotype from the early identification of FMR1 as a cause of mental retardation in boys, explaining the excess of mental retardation in boys, to Fragile X syndrome or intermediate expansion, more recently CGG expansion in Notch2 NLC, causing neuronal intranuclear inclusion disease, a disease that might not be that familiar to this side of the world, but much more common in East Asians. Some sort, I would say, Cadesi-like disease. They might not agree, but there's encephalopathy, fluctuating encephalopathy, and then dementia, headache. But CGG expansion have also been associated to neuromuscular condition, to the relevance of the Stoke, and in particular, 5 genes over the years have been associated with a particular type of muscle disease, causing distal weakness and facial or cranial nerve weakness, cranial muscle weakness. And more recently, some have been associated with CMT, and I'll come back to that. So this particular type of myopathy with distal weakness, which can be in some cases be mistaken as CMT, as it happened in one of our cases at the beginning, was described in the 70s in Japan. So prominent, here is a case, an advanced case, prominent facial weakness, dominant pedigrees. Sometimes cases are sporadic. It starts in the second, third decade, usually ptosis, but also CMT-like with distal weakness. It does progress, though, to involve facial muscle, ptosis, ophthalmoplegia, and swallowing difficulties. Oculopharyngo-distal myopathy. The rim vacuoles, intranuclear inclusion, this is a hallmark, CGG expansion generates peptide that then are translated back in the nucleus, like also the Huntington peptide. And it's similar, but not that similar to the more known oculopharyngo-muscular dystrophy, the Canadian one, commonly seen everywhere, I would say, which has a later onset. There's no real ophthalmoplegia, and the weakness in the limbs is proximal. Of course, they both have swallowing difficulties. So starting from the consideration that all these 5 genes that are not really seen at our latitudes, they all share an expanded CGG motif to cause OPDM in East Asian. We thought that there might be other genes carrying CGG expansion in Europeans causing this particular condition. So we leveraged the 100,000 Genome Project to and looked for an enrichment of large CGG expansion in myopathy cases absent in controls. And using expansion ante de novo with the outlier method, we identify our top candidate as ABCD3. And here is the estimated, everything above 50 3 nucleotides is estimated. 150 is the cutoff for the read length of short read. These are short read, but you see the estimation works just fine. So these 2 cases, the most expanded over 100,000 subjects, they do have OPDM and they have CGG expansion in ABCD3. As I told you, we performed the optical genome mapping, and you can see from the reference to the patient an insertion in that region, an insertion that is a repeat expansion. As we confirmed in multiple families, this was a discovery that was shared and done independently by Jean and Ravenscroft in Australia. So it's 8 unrelated families, over 30 individuals. We performed adaptive sequencing. This was IRA who did it in all samples, and we confirm that cases have from 120 to 700 repeats. As I told you, it does provide long read information about methylation, and pink here as opposed to purple is unmethylated, which tells us this is the promoter of the gene where the CGG is, that the gene is expressed. So I won't go in detail over the clinical features, but they had typical OPDM. I'll just show you a video of a patient that you see classical foot drop, distal myopathy or CMT, but then you see there's some more going on in the face, ptosis, ophthalmoplegia, and swallowing difficulties. So I'll also skip the pathology, but when it comes to mechanism, I like to show you that this CGG expansion, a bit like in FX TAS, are associated with upregulation. These are the 3 cases where we've done RNA-seq of muscle from ABCD3 expanded patients, and you see they have the highest level of ABCD3. So loss of function is not a mechanism here. The ABCD3 gene is transcribed and is higher compared to any other myopathy or controls, and here they're all ranked. So after HBCD3, our cohort of PDM at the national is over 50 cases now. Most remain unsolved. We found some cases with new TEP2B, which is also found in East Asian. And more recently, we found a novel candidate, which is, I would say, now confirmed. I'm going to share what's the candidate with you. Is TBC1D7, same approach. So I'm not going to repeat myself. Large expanded carrier of a CGG expansion in the promoter of better 5' UTR of TBC1D7. These genes, they do completely different things. I don't think the gene function is relevant. Lidvey in Antwerp also got to the same finding with joint data. And I can show you that this can be a quite severe disease. This patient, when I examine him, it looked like ALS, incredibly weak in the forelimbs, but with the hallmarks of a PDM as well. While this other patient is a bit less severe, but prominent fascial weakness, very prominent, ophthalmoplegia, and distal limb weakness. In the interest of time, I just move forward. So why I want to show you this example, not just to make a catalog, but because I think it really illustrates an extra layer of complexity of this CGG repeat expansion. Because I saw this family, the last patient, her, and together with her dad and mom, and they were absolutely fine. We did the PCR, we realized it was coming from the dad, we did long-read sequencing, and we realized that while she had 150 repeats, he had 270. So why is he not affected? Well, if you look at methylation data that only long-read provides, then you'll see that methylated is red in this case, and the repeat expanded containing allele in his father is completely red, completely methylated, turned off, preventing expression of the gene and translation of the repeat. While in her, it's all blue, all non-methylated, so that the transcript from TBC1D7 can undergo the downstream mechanism. Downstream mechanisms, they are largely peptide toxicity driven, and I wouldn't really call into play here RAN translation. You may know of RAN from C9, from SCA7. I don't think it's RAN translation. I think that Nicolas Chalet-Bergerande showed us pretty convincingly that the CGG expansion, they are part of upstream open reading frame, while there's a canonical AUG starting the transcription of the gene, and this is the case of Notch. There's another cognate codon or ATG just before the CGG, which starts translation that will then stop, but will produce repeat peptide, which are mostly contained in polyglycine, which will be translated back to the nucleus and probably interfere with splicing and lead to muscle degeneration, neurodegeneration. And this is kind of leading to a first shift in the interpretation of repeat expansion. We tend to look at repeat like what's the threshold? Normal, intermediate, full pathogenic. Well, here in CGG repeat, I would rather think of them as a pathogenic range, because yes, they have to be expanded enough to generate the repeat peptide, but if they get too large, then the gene is hypermethylated and this exerts a protective effect. So it's really pathogenic interval rather than threshold in CGG. And now moving back to the CMT, as I think most of the audience is mainly interested in CMT, it's worth noting that two of these OPDM genes were associated in Asians with neuropathy, and not negligible fraction, up to 10% of unsolved CMT, which would be the most frequent gene after PMP22, mitofusin, GJB1, and MPZ. So something you don't want to miss. And the type of neuropathy, I think in LRP12, it looks like really a motor predominant CMT2, while in Notch is more of a sensory motor or mainly sensory and autonomic neuropathy. But it's also true that patients with the classic need NS phenotype due to CGG expansion in Notch do have a profound denervation when they look at skin biopsy. So the nerve is involved and relevant to the field of CGG repeat expansion. So we just wonder, what about in the UK and in Europeans? Are we missing CGG repeat expansion causing neuropathy? Well, we started looking at the CGG that were associated with OPDM, and we looked at undiagnosed CMT 560 and 32,000 controls, short read data, and short answer, no. We don't think that in the UK there's any patient, here you see the cutoff of 50 repeats, no patient is really expanded, so that notch 2 or LRP12 they're unlikely to cause CMT in Europeans, and nor any of the other OPDM genes. But, these also gave us the opportunity to delve into the origin of this repeat, history, migration, ancestries. And you see that when you look at the mutable normal allele, intermediate expansion in the genes which cause OPDM, which cause neuropathy in East Asia, and you see that at population level, the size of this allele, and take this one as the 99 percentile, is larger compared to the other ethnicity and the Europeans. Notch 2, LRP12, while ABCD3, which is found in Europeans, causing ABCD3 OPDM in Europeans, is smaller in the other ethnicities. And this, I would say, really suggests how explain how the presence of intermediate allele justify the ancestry-specific propensity of this repeat with intermediate alleles to further expand in the full pathogenic range and cause disease in specific geographic region, but not in others. But the mechanisms are the same, which also holds promise in terms of unifying the development of unified treatments. And the second shift that I want to mention in this field is that is a shift in how we interpret variation in rare diseases. Because if you think about single nucleotide variants, in order to call something as pathogenic, ideally you want it absent in controls. You want it to be zero on NOMAD. But in repeats, it's exactly the opposite. And this is following the line of thoughts that I've been telling you. So Stefan has looked at the normal variability in the population from long-read data of CGG repeat expansion. And as you see that in the population, the most mutable alleles are the ones that cause disease: FMR1, DIP2B, GYPSY1, AFF, Notch. And this really means that being mutable in the control population, generating intermediate allele, is the prerequisite, not enough, to then further expand and cause disease in that population. Right. The last 5 minutes, I think I'm going to switch from DNA to RNA and see how we use long read to help interpretation of non-coding variants. People tend to do, if they can, if the labs reimburse this, to do transcriptomic. It's quite expensive. It's nice. It works well. It still doesn't give you information about isoform. We benchmarked the long read sequencing, and instead of doing a long read sequencing transcriptome, we looked at genes which we thought were the right ones, but we had to prove pathogenicity for some VUSs that would have otherwise remained VUSs in non-coding region. This is amplicon based. We did a PCR of the full transcript from the 5 to the 3' UTR, and we sent it for nanopore sequencing, and we saved money. It just cost roughly £10 per sample to sequence. And here's some example. Is a patient with Behr syndrome, optic atrophy, ataxia, severe neuropathy, as you see. And she had a variant, interestingly enough, after the last coding exon. And so difficult to make it pathogenic is after the stop. It was a deletion of 2 nucleotides. It was predicted to alter splicing, so we amplified the old transcript. And what we realized, yet it's homozygous from consanguineous parents, is that particular variant after the stop codon it does lead to skipping of the last exon, so leading then to frame shift in 26% of the read. If you do NMD inhibition, it increases. And we also realized that, splicing is not like one event only, and that's it. It can be more complex. We also saw other reads that were negatively impacted by the retention of the last intron. So they probably both contribute to leading to a non-functioning protein from the both alleles. Same on this patient with the distal type of myopathy. This was a bit easier. It was in the splicing region, splicing site, -5. PROXD1, they got this blue sclera as a hallmark. And again, in this case, at full transcript level, we are able to show that we confirm the predicted incorporation of nucleotides leading to nonsense mediated decay. Finally, again, coming back to CMT genes, GDAP, 23 nucleotides variant next to the splice junction. Sometimes people limit it to 22, so it's technically just outside the splicing region. And here we prove that, again, he alters splices, incorporate nucleotides, leads to degradation, nonsense mediated decay of the transcript. And this is, again, homozygous. And we were able to provide the final confirmation. Right, so long read is great. Optical genome mapping is good too. Very large structural variant are very suitable for OGM. Here is a DMD case that was negative of all the previous testing that was shown to have an inversion, disrupting the reading frame. Very promising application in FSH from, and this is work from my colleague Enrico Bugiardini. So, I mean, there's room for implementing OGM. We have 2 ongoing projects with the support of CMTA and AFM Teleton to take forward the diagnosis in unsolved CMT patients or neuromuscular patients. If you have patients with missing second variant, we're very keen on working together of those. We have separate lines of interest in axonal CMT cases, OPDM, and sensory neuropathies negative for Canvas. So very happy to collaborate on interesting cases. A family. Last 2 slides are, as Cleopas said, probably, and that's his judgment. My main contribution to science was the identification of RFC1. Indeed, this is an incredibly common disease, biallelic expansion, different repeat motif from the reference. 22% of ataxia, 60% of ataxia and neuropathy, 30% of isolated neuropathy. The disease progresses from asymptomatic, maybe there might be something in this very young guy, cough, sensory symptoms or not, ataxia and walking aids. And I think 1/3 of these people with a sensory neuropathy, they have it, and they should be tested as much as they tested for glucose vitamin deficiencies. Complex repeat, multiple units, so still probably suboptimal diagnostic tests, but very common. So, advances in molecular genetics here are highly needed. The repeat is spread over centuries, millennia, across the whole world. So it's not ancestry specific, but when you look now cross-sectionally at the repeat expansion in the population, you see the RFC1, these are European, is by far the most common repeat, much more common than Friedreich, and of course of any dominant repeat expansion. So here we are at the phase that we want to understand better the mechanism of RFC1 degeneration. We think we worked pretty hard over the last 5 years to provide evidence of loss of function. So this is a talk about genetics. There's no time about that. But for those interested, the paper is on bioRxiv and being reviewed. If you're interested in mechanisms, I have a ERC grant to do a bunch of interesting things to try and rescue this loss of function. Please touch base. And the work, as said, on this can be done in London. Some of it, for those that like warmer weather, could also be done in Milan, particularly if you like food, fashion, that would be good, but there's a lot more going on in Milan as well. London needs no introduction. Final initiative will never stop, will probably never stop. We'll try to identify genetic modifiers of this condition of RFC1. So if you have cases, please do touch base, but also TTR amyloidosis. We'll present at the next PNS conference and preliminary results at the ISA conference. Striking variability for a gene with one mutation, and we want to ideally get to the bottom of it, or at least try. Conclusion, I think new technologies are enhancing diagnosis of neuromuscular diseases. They're helping patients, but we clinicians need to know when to use them. And, repeats, missing second variant, phasing of the alleles are good, places to start with. But still, the interpretation of variants, and particularly of non-coding variants, is still a major barrier to provide diagnosis, and this is something we all want to work on. And a big thank you to many people from my group. Some are still here, there's new ones, some have left, and to the funder which made all this possible, and particularly CMTA and AFM Teleton, very relevant to this talk. And thank you everybody for listening.
Thank you very much, Andrea. It was an amazing tour over the exciting developments in genetic discovery and the amazing work that you have been doing.
Thank you.
I don't know, Elizabeth, if we have any questions in the chat or in the Q&A.
We have a little bit of time. You can go out of 5 p.m. It's okay.
Yeah, I had 48 minutes. I thought we had to do 45, but I hope it's still all right.
Yeah, we have 5 minutes for.
Yeah. I've been 3 minutes late.
So I think, my take home here is that there is really a lot of variation depending also on the ethnic background, the methylation status. So the picture becomes more and more complex. And so the question for the clinician is, what is the sequence of technology use to, finally reach a diagnosis? And I think the other important point was the phenotypic complexity. See all these mixed phenotypes and all the overlapping phenotypes, and then it's really important to know which genes could be really relevant for the diagnosis.
Yes. So in the field of CGG repeat expansion, I think that we're still learning. This is what the first thing to full disclosure, we're still learning. And I think we are at the stage that there are a lot of highly reliable short read data. And there are good tools, bioinformatic tools, to use this data. So I think, at least in our hands, the starting point where the analysis of this short read data to get some signal, and then to further confirm it using long read sequencing. So I think that what we're doing at the moment is leveraging large databases. And will it be conclusive, the data from short read? No. You need to do more, you need to do long read, and then you can either confirm or reject this pathogenicity. Again, as always in medicine and science, making a claim that something is pathogenic, is relevant, is true, has to go through validation, replication in independent samples. And here having good clinical data is essential to compare phenotypes and to show that the same expansion in the same gene can lead to a similar phenotype in independent families. So this is how we kind of confirm these genes. It's also true that there might be more limited, site-restricted variants, patient with only distal myopathy, only with ptosis. And I think that now UK Biobank and other initiatives of that sort can tell us whether I would say, claiming that we have a new repeat expansion causing disease has to go at this stage. We may want to go through at this stage like classic segregation in families to get a more insight on the full breadth of the prevalence and phenotypic variability. We may want to look at population data and see whether some of these repeats are associated with particular forms of, more common complaints from ptosis, distal weakness, which is something we're doing.
And I gave this, the population data has to be in the context of the ethnic.
And it has to be in the context of the ethnicity, absolutely.
And I have another question. What do you think is the role of the functional studies in this genetic?
Yeah, I think functional studies in the CGG repeat expansion are coming. I think there's good data from my collaborator Nicolas in Strasbourg, but also excellent work from our colleagues and friends, some of them, where I know them, in China and Japan, modeling the toxicity. As said, I think the unifying mechanism is translation of the repeat. Translation of the repeat, formation of nuclear aggregates that are quite unconspicuous on the muscle biopsies. So I would trust more like P62 markers rather than looking at these eosinophilic nuclear aggregates. But those aggregates, like the Huntington nuclear aggregates, they're toxic. The toxic and probably disrupt splicing, transcriptome, and via transcriptome, oxidative stress, and muscle degeneration.
So the RNA long read is crucial for.
It would be interesting, no one has done it, to see the full breadth extent of the changes of the splicing pattern. They've looked at more specifically some genes. They might have done it with RNA-seq. This is something we're planning to do. We're planning to apply for that, to do that, and to rescue it, because we think those genes are tolerant towards haploinsufficiency. And so a possible stability strategy we want to pursue is downregulation.
Okay, great. I don't know, Elizabeth, if we have any other questions from the audience. I don't see anything in the Q&A.
Yes, there's another question.
I cannot see the questions.
The Q&A?
No.
Maybe I got a chat. I can open a chat. Let's see. I know, but chat is just
it's not in the
admin.
There should be another box, but
Q&A.
Yes,
Q&A. Yeah, I can read out the. Soria is asking, can it happen without any family history of vitamin deficiency? It depends on what you're referring to. Which part? I'm not sure.
She asked to unmute herself, if you may, Andrea.
I have to mute myself.
Not you. I will unmute Soria. Soria, you can talk if you wish.
Soria, yes, unmute yourself.
Hi, good evening.
Hello.
How are you? Thank you for this lovely evening. I have a CMT HMS type, but I don't have a family history and I didn't have any vitamin deficiency. I'm waiting for my genetic test to come out. But can it happen without having any family history?
You mean SORD?
I don't know yet. I just know that I have CMT.
So you have a patient with CMT and you're asking me, is it possible that this guy has CMT despite the fact that neither of the parents or brothers has a similar condition?
Yeah, that's right. Not even anyone in the family have it.
No one in the family. Yeah, the answer, Cleopa, I see is nodding. Yes, it's absolutely possible. because, for two reasons. First of all, it could be a recessive form of CMT. And so one mutation from mom, one from dad, they're healthy. And but with two mutations on two separate alleles, like in SORD, you get the disease only in the offspring. Particularly at our latitudes, now families are not that big anymore. You don't have 10 siblings. So having only one, it's it's absolutely compatible, right? Otherwise, for those recessive in a large family, maybe you presume you might have two, but it doesn't have to be. It can be only one. Another reason is that the mutation may have occurred de novo. So it could be a dominant disease in a way, and the mutation has arisen de novo from mom or dad. And the ones that you have is the first one who has the mutation, and it may well pass it on to the next generation, but you don't know. And the third reason is variable penetrance, particularly with repeat, but also with single nucleotide variants. We increasingly realize in CMT that, there might be striking variability, and one of the parents might have the variant and not the phenotype. That's why in dominant families we tend not to test unaffected, because we wouldn't be able to rule out pathogenicity based on presence of the variant in someone unaffected, because it might be only reduced penetrance, as in the case of CGG in that family in TBC1D7. He had the repeat expansion, it was just hypermethylated.
Okay, thank you very much for
thank you. A couple of questions if we still have some time. I see from Davide Bareson here. Great talk. Many thanks. Can you comment a little bit on role of methylation?
Yeah, I think methylation is something really that we're starting looking now in the field of CGG repeat expansion. It might have a broader effect, but the CGG, CPG islands are methylated. And the promoter where the CGG repeat expansion sit have also additional CPG island. So the repeat can be methylated, the flanking CPG island can also be methylated, and that would make the chromatin more compact and less amenable to transcription. So that is what causing downregulation of the gene, turning off the gene, a gene which otherwise would bear a potentially toxic polyglycine forming stretch of poly-CGG. So this is what we know from the CGG. But again, it might be that methylation modulates expression at other sites, other mutation, and we're not systematically looking for it, but long read will allow that.
Okay, thank you. Maybe one more question from Colleen Scales.
Yeah.
Listening, so she says she has a family of CMT sufferers who are on any recommendation on how or which testing approach for a family with one parent as known sufferer who has since passed away, and then there are two siblings and grandchildren now displaying symptoms.
Any recommendation now which testing approach for a family with one parent as known sufferer who has since passed away, children and grandchildren now display symptoms, teenager? What's available in terms of effective testing and cost in the UK? Okay, Cleopa, you missed an important part of the question, which is a family of CMT sufferers who are under Queen Square under a certain Mary R. So, which I think I know the person. I think this family is in excellent hands. So, but the question is still well placed. I think unless the DNA from the grandparent affected is available, was stored, and there is, consent in place to retrieve it and use it, in this case, I would focus on the affected only in the family. And if possible, I think you have two siblings and grandchildren. That depends on how much the NHS genomic medicine will allow you to sequence. If they allow you to sequence, usually it's 2 to 3. Usually they don't say no to 3. So I would sequence probably the most distant of the family. So if you have cousins, those are probably the most informative, and you're going to try to find the shared variant in both or in the three affected. And the more distant, the more informative, because they've undergone more recombinations.
Okay, thank you. So
I hope this is what, has been done.
there is one last question. Can you see that about patients having mutation but not being sick?
So, an anomaly has been found. Is it possible the candidate is not actually sick? Sorry. Thank you so much. I have a general question. After a genetic test, even if an abnormality has been found, is it possible that the candidate is not actually sick? For example, in case where PMP22 was found to be double in a genetic test, is it possible that the patient does not have CMT? Yes. So in this particular example, in this particular one, unlikely. Usually CMT1A PMP22 duplication is highly penetrant, highly penetrant, at least neurophysiologically. People will tend to have slow conductions. Then the phenotype may be much milder, as you say, and it all depends when are we examining this subject. But, taken at genome-wide level, what you are mentioning is one of the main challenges of the genome era, because we do find incidental variant, potentially pathogenic, and sometimes in genes associated with cancer, neurodegeneration. And that is a byproduct of the test. You were testing for something, but then the lab returned to a result about something else. The patient doesn't have it. And I think at that point, it's crucial that before starting the test, a clear conversation whether the patient would like to be informed about incidental finding, actionable incidental finding, any incidental finding informing on prognosis. So this discussion ideally happens before the whole genome sequencing.
Great. Thank you very much, Andrea, for answering all the questions, for the great talk. Thank you all for joining and participating. And we'll see you next Thursday. We have another great talk by Mary Riley on biomarkers and clinical outcomes. So please join again. It will be 1 hour later, though. Please keep that in mind.
Perfect. Okay, I'll try to join.
Thank you.
With a pleasure. Thank you so much.
Have a nice evening. Thank
Goodbye.
Thank you.
Bye.
Goodbye. Have a nice
Bye,
evening.
everybody.
Bye-bye.